Today, we are examining one of the most important communication points in the nervous system, the neuromuscular junction. This is the specialized synapse where a motor neuron communicates with a skeletal muscle fiber in order to produce movement. One of the most important ideas to understand right away is that the neuron and the muscle do not physically touch. Instead, they are separated by a very small space known as the synaptic cleft. In this diagram, the enlarged end of the neuron is called the axon terminal, sometimes referred to as the terminal button. Inside the axon terminal are many small membrane-bound sacs called vesicles. These vesicles are filled with a neurotransmitter called acetylcholine. On the muscle side of the junction is a specialized region of the muscle membrane called the motor end plate. This area contains a high concentration of receptor proteins. When an action potential arrives at the axon terminal, it triggers the opening of calcium ion channels. Calcium ions move into the neuron, and this entry of calcium acts as a signal that causes the vesicles to fuse with the neuron membrane and release acetylcholine into the synaptic cleft. Acetylcholine then diffuses across the gap and binds to receptors on the muscle membrane. This binding opens ion channels and allows sodium ions to move into the muscle cell, creating an electrical change that triggers a muscle action potential. This process converts an electrical signal into a chemical signal and then back into an electrical signal, allowing movement to occur. This slide focuses on what happens when a signal reaches the end of a neuron. Along the neuron, signals travel electrically as action potentials. However, the signal cannot continue electrically across the synaptic cleft because there is no physical connection between the neuron and the muscle. Instead, the nervous system uses chemical messengers known as neurotransmitters to carry the signal across the gap. At the neuromuscular junction, the neurotransmitter involved is acetylcholine. When acetylcholine is released, it moves across the synaptic cleft and binds to receptor proteins located on the muscle membrane. These receptors are ligand-gated ion channels, meaning that the channel opens when a specific chemical binds to it. Neurotransmitters can have different effects depending on the type of receptor they bind to. Some neurotransmitters are excitatory, meaning they increase the likelihood that the next cell will generate an action potential. Others are inhibitory, meaning they decrease the likelihood of firing by preventing depolarization or making the inside of the cell more negative. At the neuromuscular junction, acetylcholine is excitatory because it causes sodium ions to enter the muscle fiber, pushing the membrane toward threshold and triggering contraction. This synapse is therefore a control point where the nervous system determines whether a muscle will contract or remain relaxed.
This slide expands our understanding of neurotransmitters by placing acetylcholine in a broader context. The table on the slide shows several major neurotransmitters and their general functions. Acetylcholine is especially important because it is the primary neurotransmitter used by motor neurons to control skeletal muscle contraction. In this role, acetylcholine is typically excitatory. Other neurotransmitters serve different purposes in the nervous system. Dopamine is involved in motivation, reward, and movement control. Gamma-aminobutyric acid, often called GABA, is the primary inhibitory neurotransmitter in the brain and plays a critical role in preventing excessive neural activity. Glutamate is the most common excitatory neurotransmitter in the brain and is essential for learning and memory. The diagrams on the slide show the general process of synaptic transmission. Neurotransmitters are released from vesicles, cross the synaptic cleft, bind to receptors, and change ion movement in the receiving cell. It is important to remember that neurotransmitters do not carry electricity themselves. Instead, they influence whether ion channels open or close, which then determines whether the next cell will produce an electrical signal. The effect of a neurotransmitter depends on the receptor it binds to, not just the chemical itself.
This slide serves as an opportunity to review the sequence of events at the neuromuscular junction and prepare for what happens next. The process begins when an action potential arrives at the axon terminal of a motor neuron. This causes voltage-gated calcium channels to open, allowing calcium ions to enter the neuron. Calcium triggers the release of acetylcholine from vesicles into the synaptic cleft. Acetylcholine crosses the synaptic cleft and binds to receptors on the motor end plate of the muscle fiber. This binding opens ligand-gated ion channels, allowing sodium ions to enter the muscle cell. The movement of sodium ions causes a local depolarization called an end plate potential. If this depolarization reaches threshold, a muscle action potential is generated. That muscle action potential then spreads along the muscle membrane and leads to muscle contraction. It is equally important that the signal stops at the correct time. An enzyme called acetylcholinesterase breaks down acetylcholine in the synaptic cleft, preventing continuous stimulation. This ensures that muscle contraction is precise, controlled, and only occurs when intended.